Yes, little champions, how are you all? I hope everyone who is watching this video is doing perfectly good, great, happy and healthy. I know KCET is there, PU2 board exam is there, but here we are with a very important master plan, that is the Masterstroke, the one-shot videos for all the important chapters up over here. And starting with biology session today, at least I saw some of you asking to me that, ma'am, when is the Masterstroke starting for biology? So I was kind of happy because mostly I'm sad seeing that there is no KCET crowd for biology. I know you are somewhere hidden out there. Please, my dear children, respond to me, talk to me in the comment section, tell if the session was good or bad or whatever is there, because even I need some amount of motivation, right? So yes, so it will be a one-shot video, it will be quick. I'll try to wrap up quick, but I'll include all the important points which would definitely give you answers. Now tomorrow there will be a live session at 11 a.m. So you have to be there with me for the live, and the live is obviously so fun. So we will enjoy and we will study really well. So let's get started, okay? So I just want one thing from you, that is take out your NCERT. Whenever I talk about something, just go through it, that's all, okay? Just listen to me all ears, just know that it's there in the NCERT. I just want you to know that, okay, this is there over here in the NCERT, that's all, yeah? So let's get started with the first thing. Okay, so who is known as the father of genetics, my dear children? It is Gregor Mendel. And the first question that is very much important is, for how many years did he actually do his experiment? So for 7 years he did his experiment. You have to know the year as well. It's from 1856 to 1863. What? 1856 to 1863, my dear children. He did his experiments, and for that, what did he choose? He chose Pisum sativa, or basically garden pea. Okay, so Pisum sativa is not being mentioned over here, so I'll just write it down for you. It's Pisum. Is it visible? No, it's not visible, so let's make it in black. Pisum sativa, or basically garden pea, was being used. Now, how many varieties of pea were being used? So 14 true breeding pea plant varieties were being used. So all of them were in pairs. So we say 7 pairs, or we say 14 true breeding pea plant varieties. Okay, now over here, what were the characters that he chose? So obviously, as I said, 7 pair of plants he chose, right? So 7 pairs of contrasting traits were also there. And what are those traits? Stem height, flower color, flower position, pod shape, pod color, seed shape and seed color. So over here you might have seen pea. Pea, how does it look? So it has a covering like this, inside which you have the pea, right? So this thing that you see over here, this is known as the pod. So depending whether the pod is inflated as in it is full, or it is constricted as in it is dry, or things like that, on the basis of this also we have 2 contrasting traits for each character. For stem height, you know there is tall and there is dwarf. For flower color, you have violet and there is white. Okay, there are violet flowers also, there are white flowers also. Flower position, it can be either like this in the axial position, or it can be over here in the terminal position, not in the axial position. That is what you mean by axial and terminal. Now pod shape can be inflated, as in it can be full, okay, proper one like this, nice one, okay, or basically it can be constricted like this also. Now pod color, it can be green or yellow. Seed shape, if you're talking about the seed shape inside, it can be basically round or it can be wrinkled like this. Now seed color, it can be yellow or it can be green. Now the next question that will be coming from this particular thing itself is that, what is the dominant one, what is the recessive one? So they won't give you a direct question what is dominant, what is recessive. They will give you a set of character that is probably tall and then axial, inflated, green and then violet, and they will ask you which among them, okay, which among them is not a dominant trait. So you should be knowing which is dominant, then only you can answer which is not, right? So over here, the first words, whatever is written, they are all dominant. So tall is dominant, violet color flowers are dominant, axial flowers are dominant over terminal, inflated, okay, the full one is dominant over the constricted one, the green color pod color is dominant over the yellow one, which is why when we go to bazaar and all, we take the green color ones. Have you ever seen yellow pods? No, right? Now seed shape, if you're talking, round is dominant, wrinkled, obviously we do not want, that will be recessive. Seed color, if you're talking about that, then yellow is dominant and green is recessive. So this is something unique. We usually eat green peas, it is green in color, but actual color which is dominant is what? Yellow. So this is a unique way to remember. Mostly we pick green, but actually it is what? The yellow one which is dominant in trait. Cool? Works? Cool? So these 7 years, 14 true breeding pea plants and 7 pairs of contrasting characters were there. Okay, so basically how did he count 14? 1, 2. Tall plant, dwarf plant. Violet 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14. Got it? Please remember this also. So what is important is only been put up here. Nothing is there to confuse you guys. So you can see a lot of things in your NCERT which is there. I won't suggest you to just put on your head into it as of now. You just have some days left and you have to concentrate on what's important. Right? Now, when he did an experimental cross, so what he took is that he took two pea plants. One was tall and other one was dwarf, and then basically he crossed both. Okay, he crossed both. And what did he call them? He called them pure breeding tall. Okay, pure breeding tall and pure breeding dwarf. What do you mean by that? Basically, that means both the alleles were, my dear children, homozygous. They were same. It's capital T, capital T. In the case of dwarf, it was small t, small t. What do you call this condition as? You call this condition as homozygous condition, where both the alleles are, my dear children, going to be the same things. Fine. Now, you have tall plant, you have dwarf plants. Then he crossed them. Okay, he crossed them. So in this case, if I zoom this in, if this is the case, capital T is over here. In the gametes, the plant is going to produce gametes. In the gametes, what will be there? Again, either this chromosome can go or this chromosome can go. Either way, it will be capital T that will be going. So I have written capital T, capital T. In the case of dwarf plant, again small t can go or another small t can go, because both of them, they are pure breed, or basically what do you call, homozygous for the same traits. Now going ahead, when you cross them, capital T, when you cross it, you get capital T small t. This you cross with this, again you get capital T small t. This you cross with this, capital T small t. This with this, again capital T small t. All of them, my dear children, are capital T small t. Now what's the thing about capital T small t is that, suppose it's like this. Okay, this is a condition. Now, is it homozygous? Are the alleles same? No, it is different. Okay, now in this condition, one will be expressing over the other one. The one that is dominant, the one gene that will be producing more proteins, that will be obviously visible. So, obviously, this allele over here is more dominant over the small t. And this condition you call as heterozygous, because it's not the same anymore. The alleles are not same anymore, and over here one is dominant over the other. So, all of them, they are capital T small t. That means all of them have to be tall. They are tall. Now, this is a physical appearance, that is tall, dwarf and things like that. So, what do you call this? This is known as the phenotype of the plant. This is known as the phenotype of the plant. What is the phenotype, my dear children? Whether it's tall or dwarf, the physical character. Now, what is genotype? Genotype is the chemical composition, or basically the genetic, not chemical composition, the genetic makeup. Over here it is capital T small t. This is what? The genotype of the plant. What is the genotype of this plant, my dear children? This is the genotype of this plant. What is the phenotype? It is tall. What is the genotype? It is small t, small t. What is the phenotype? It is dwarf. Got it? So now I have capital T, small t. What I am going to do is, basically what did Mendel do is, he crossed the progenies among themselves. And then what did he find? Let's see. So this time, story is going to be a bit different. So in the gametes, it can be either this chromosome, that is small t, or the capital T chromosome. It's a pair of chromosome on which you have two alleles. So it looks almost like this. Okay, it looks almost like this, where you have capital T over here and small t over here. So from a pair of chromosome, only one chromosome is going to go. Only one chromosome is going to go. That is how gametes are being formed. Half the number of chromosomes are going to go over here. So there it can be this, or it can be this in the gametes. So let's see. So capital T or small t over here, again capital T or small t from this plant also. Now, if suppose the gamete which is carrying capital T fuses with another gamete carrying capital T, it will be capital T, capital T. Suppose this and this fuses, it will be capital T, small t. This and this fuses, my dear children, it will be capital T, small t. This and this fuses, it is small t, small t. So as you can see over here, one among them is homozygous. If we talk about genotypic ratio, you see capital T, there is only one. Small t, there is only one. What about them? Capital T, small t, the heterozygous, if you see, they are 2 in number. So what is the genotypic ratio? Genotypic ratio is 1:2:1. What is a phenotypic ratio? How do they appear? So this is tall. What about this? This is also tall, because capital T is there. It will be dominant over the other one, so it will be tall. This also will be tall, my dear children. So if we talk about phenotypic ratio, how do they appear? So for me, who do not know anything about genetic makeup, they all appear, 3 of them, they appear to be tall, and 1 of them, they appear to be dwarf, my dear children. So the ratio will be 3:1. So this is what? This was the first generation that he got, where everyone were what? Capital T, small t. This is F1 generation, filial 1 generation. Over here, all of them basically you see different type of plants are being obtained over here. This is F2 generation. Okay, this is F2 generation. You have to know about all this ratios and things like that. I have already discussed as of now. So in F1 generation, if you have seen, you are only seeing the dominant character. You do not see the recessive character. So you feel that the recessive character is not there at all. But in F2 generation, the recessive character pops up. Okay, that's the beauty of genetics. Now this is a monohybrid cross because you're talking about just a single trait, that is the stem height. Okay, and hybrid because you're getting a new type of plant that is having capital T, small t. It is a hybrid now. It's not pure breed. Okay, so monohybrid cross stands for that. Now going ahead, my dear children. Okay, so there is something that is known as test cross. What do you mean by test cross? Basically, I saw a flower is there and the flower is violet in color. The plant is there, the plant is having violet color flowers. Now I'm not sure, now violet you know is a dominant trait, but I'm not sure whether the violet, suppose I am saying capital W stands for violet. But I have to be sure whether it's like this, it is having capital W, or whether it is like this. That is whether it is homozygous violet or it is heterozygous violet. Because if it is heterozygous violet, then if I fuse other plants, my dear children, I will be getting what? White color ones also, because there is a white color gene also hiding over here. You saw in the first generation, dwarf one was hiding, right? So similarly, I just want to confirm. So how can I confirm that? Basically, I can fuse both of them. The plants which are, whatever plant is there, I can fuse it with a recessive parent plant. I can fuse them with a recessive parent plant. Now what is the difference? What difference does it make, ma'am? So I'm going to show you over here. So this is the recessive parent that I am having over here. This is the recessive parent I am having over here. And supposing if it is capital W, the violet flower is basically plant is having capital W, the gametes will be like this. Now if I fuse capital W with small w, all of them, the combination will be capital W, small w. All of them, my dear children, since capital W is there, it will be dominant over this and all of them will be violet in color, all the plants. But suppose the plant which was there, which was appearing to be violet, its genotype was like this. Now when I fuse, it will be capital W, small w, it's fine. Again capital W, small w, it's fine. But what about this? This is small w. The other allele was small w. And now when this small w fuses with that small w, you get small w, that means it will be white in color. Again this fuses with this, you get white flowers. So you get to know that this is the genotype of this particular plant. Otherwise you won't be able to understand. Okay, now this is known as test cross. So in a typical test cross, an organism showing a dominant phenotype. Okay, organism showing a dominant phenotype, that is what? Violet color flowers, is crossed with a recessive parent instead of self-crossing. So it is crossed with what? A recessive parent instead of crossing among themselves. There is no use of crossing among themselves. So this is known as test cross. So you should be knowing in test cross, you are going to take what? A recessive parent, my dear children. Will you remember it? Yes? And if at all you are getting bored, do let me know in the comment section that, ma'am, please make it more interesting or things like that. I am trying my maximum to make it more positive, more engaging, fully energetic. But if at all you need, let me know. And if you think that the session is going really well, then also I need a little amount of motivation, as I said. So like the video and also comment down. Okay? Also your doubts. Now, depending on the monohybrid cross, Mendel gave two laws of inheritance. Okay, two laws of Mendel is there, which is law of dominance, my dear children, and law of segregation. I am going to explain you here out to me really well. So, law of dominance says traits can be controlled by factors that occur in pairs. So he said, if you're talking about height basically can be controlled by factors. He was not known, he didn't know about genes and all. Okay, he was the first scientist to work on it probably, right? So obviously he does not have any idea about genes and genetics and a lot of things. Still he tried and he got to know that basically there are certain factors which are responsible for traits, that is height or whatever is there. And these factors are going to occur in pairs. That means you are talking about capital T and small t. These are the factors and they are going to occur in what pairs? Or basically capital T or small t, whatever you want to take, you can take. Now one allele is dominant. In these factors, this different type of factors, you call each of them as allele. So this is allele 1 and this is what? Allele 2. Now, one allele is dominant and the other one is recessive. Okay, according to law of dominance, he said that one allele in a pair of factor, if the factors are different, not like this. If the factors are different, one will be dominant, other will be recessive. Explains why one parental trait appears in F1 generation and then reappear in F2 generation. So, according to this law only, he was able to explain that why there is only one character that is being seen in the F1 generation. All of them were tall, but in the next generation, he saw dwarf plants also because of the same reason, because there was no one to dominate the dwarf one, which is why it expressed itself. So this is a law of dominance, my dear children. Now what is law of segregation? Law of segregation says that alleles do not blend, they do not come together, so there is no medium heighted plant according to him. So both reappear unchanged in F2 generation. So during gamete formation, allele pairs segregate, so each gamete gets only one allele. That is if you are talking about one pair of allele, my dear children. Okay, now when you have to, this is the genotype of a parent. Now when this parent has to form gametes, it is saying that from a pair of alleles, one pair will be going to one gamete, sorry, not one pair, one allele will be going to one gamete and another allele will be going to another gamete. It's not like both of them are going to come together. So during gamete formation, alleles pair separate so that each gamete gets only one allele. It gets only one allele. Okay, so this gamete has one allele. Now the another parent with which it will be fusing, it will also give a gamete, and both these gametes are gonna fuse and they are going to make up, suppose they gave small t, so they are going to make up a pair again. So homozygous parents produce identical gametes. That means if you are talking about these type of parents, capital T, they are going to, whatever you do, they are going to give same type of gametes only. It will be always capital T, right? Whereas heterozygous gametes are going to produce heterozygous parents are going to produce two type of gametes. That is what is meant by law of segregation. That means alleles not blend. Okay, whenever you want to make gametes from a pair of alleles, both of them will segregate, both of them will separate, and only one allele will be going into each pair. That is what is meant. Okay, cool. So these were the two laws of Mendel, or basically Mendel's law of inheritance, which was given. There is one more law which will be talked about after we speak about the dihybrid cross. So this is a monohybrid cross. We are talking about just one character, right? So let's go ahead. Now Mendel gave his theory, but do you think he was exactly correct? No, my dear children. So there were certain exceptions to his cases as well. So the first exception is incomplete dominance. So there were certain alleles which are not completely dominant. I saw over here capital T was completely dominant over small t, but this was not the case always. And where did he see it? You have to remember the name of the plant. In snapdragon plant, or basically you call it Antirrhinum species. Antirrhinum majus is the name. Okay, Antirrhinum majus is the name, my dear children. In this he saw incomplete dominance, and it was kind of really crazy. I'll tell you why. So look at this scenario really well. Okay, over here he had red flowering plants, that is snapdragon. So he assumed capital R is dominant, so which is why both of the he took red plants which is having capital R. Not he, basically there were exceptions that were found. Now white one were having small r smaller, since white one is a recessive trait. Now whatever you do, the gametes will be carrying capital R only, because this is a homozygous condition. Here also the gamete will be giving you smaller. Capital R and smaller, whenever it comes together, it will be giving you capital R smaller. Now the thing is, this capital R should be dominant over this and the flower should be red in color. But what were you getting? You were getting pink color. Why, my dear children? Because just suppose, probably this might have not been completely dominant over white, which is why there is a mix of pink over here. Now they were kind of astonished and they kept on going. So again selfing was done. Again the plants which were having capital R, small r, they were crossed among themselves, and then you got something like this. So you have capital R, small r, again you have capital R, small r, my dear children, right? So when you fuse, let's see what happens. So when capital R was fused, you got capital R. This and this you got capital R smaller, this and this you got capital R smaller, again this and this you got smaller. Now the scenario was changed. There also when we did the genotypic crossing and things like that, okay, we found that 1:2:1 is the ratio. Okay, there also capital T was there, capital T small t was there, and small t was there. So until here it's fine. What about the phenotypic ratio? Again, the red one which was not seen over here in F1 generation was seen over here. You got red plants again. One of them, the phenotypic ratio, the appearance of one of them was red, my dear children, which were having both the capital R. The ones that had capital R, small r, they were showing incomplete dominance, which is why they were still pink in color. And the one that is having small r, they were white in color. So over there it was 3:1 ratio, phenotypic ratio, but here it is 1:2:1. So this is one exception to Mendel's law, where he said that according to his law of dominance, he said that one allele will be completely dominant over the another one, but here it was not the case. So R was not completely dominant over small r, and this made it possible to distinguish what? Capital R, small r as pink from capital R, that is red, and then white. Cool. So remember the name of the plant, and yes, everything, snapdragon, antherinum, and this is the thing you have to remember, the phenotypic and genotypic ratio as well. Case 2. There is codominance and multiple allelism. Again, both of them, if suppose capital T and small t, both of them are expressing together, that becomes odd, right? So over here, they saw, the people who were working on it, they saw that basically two alleles are expressing it themselves together. How can that be possible? Basically, this is known as codominance. Both of them are equally dominant. No one is saying that I am going to give up. Anyways, you have to show my character also. So that was being seen, and that was being seen in what, my dear children? It was seen in blood group. And blood group is also an example of multiple allelism. So seen in blood group, where what happens, the plasma membrane of the red blood cell, so you know RBC has a plasma membrane, has sugar polymers that protrude from its surface, and the kind of sugar is controlled by the gene. So basically, suppose this is an RBC, okay, just a second. Suppose this is an RBC. So there are sugar polymers which are coming out, protruding out like this. Now what type of sugars are being there? It's being controlled by the genes coding for it. Now the gene which is there, it has 3 alleles, IA, IB and small i. Okay, these were the 3 alleles. But when IA and IB are present together, they both express their type of sugars. You didn't understand? I am going to tell it. So listen to me carefully, okay? So over here, I am saying there are 3 alleles. IA, this is 1, IB, this is 2, and there is an allele, or basically a gene, that is I. What happens is that, suppose I want A blood group. A blood group will be there when both the alleles are carrying AA like this. In that case, it will be A. Or probably when one is carrying A and the other one is basically carrying I. Simple I, that is for O. Basically, why it is still A blood group? Because this might be dominant, completely dominant over small I, that is the allele for O blood group. Okay, so you got to know one thing, that A is what, children? Dominant. A is dominant, and basically this one is what? Recessive. Now, talking about B blood group. Again, a person should be having both the alleles like this, or the condition can be one can be like this and other can be like this. Again, this is also dominant over this, which is why it is getting expressed. So B is dominant. B is dominant, and what? Still small I is what? Recessive. Talking about O blood group. O blood group is seen when both the alleles are like this, in the recessive condition, just like how we were seeing DOF, no? In DOF, you need both the alleles to be same. So over here also, the allele has to be looking similar, they have to be homozygous. Then only the O blood group will be there, because it's a totally recessive allele. Now you know there's one more blood group, that is AB. And how do you get that? Basically, when one gamete has IA and the other gamete contributes IB, both of them become co-dominant and they express themselves. You get it? So how many type of genotypes do we have for the blood group? 1, 2, 3, 4, 5, 6 genotypes we have, as you can see over here. And how many alleles do we have? 1, 2, 3 alleles we have, and how it is contradictory to the law of Mendel. Basically, it shows co-dominance. Okay, so this is an example of multiple allelism. Do not forget that. I hope you understood this now. Okay, so this is a chart given for it. Just go through it. It's the same thing that I said, and it will be really important. So when both IA and IB are present together, they both express their own type of sugar, which is why you say it is co-dominant. Cool? Please remember. Chalo. Now, talking about the monohybrid cross thingy is done. Okay, so in your book there is one more example for multiple allelism as well as this, that is about the starch synthesis. You just go through it Okay, if you see just after the blood group thing, there is a paragraph given, just go through it. It's not very much important, questions have not been framed yet for KCET, but we can expect any time, but again as I said, not very important. Okay, now we have Mendel's dihybrid cross, my dear children, where until now we were talking about just one character at one particular point of time, that is what say violet white, tall dwarf or things like that. But here we are going to talk about two characters, and what are the two characters over here? Let's see. So Mendel, he basically took round plants and round seeded plants with yellow seeded plants. Round and yellow were together and wrinkled and green were together. So a plant was producing a seed that was round and yellow, another plant producing seed that was wrinkled and green. So round is dominant, which is why I say capital R. And again yellow is also dominant, so I am taking pure breeding plants, homozygous plants. So it will be capital Y. Still here it's small r and small y. Now what are the gametes that will be formed? It is capital R, capital Y. Now you might be thinking how? So for that confusion, for taking away that confusion, I will tell you easy trick. Always think of it in a sense of chromosomes, my dear children. So suppose you have a chromosome like this, a pair. So this is what? Capital R and capital Y. This is the scenario. Now, I told you from a pair only one allele will be going, that is law of segregation. So either this will go, either this will go. Whatever you do, it will be capital R only that will be coming. Here also either this can go, either this can go, it will be capital Y only that will be coming. That means one chromosome capital I, another chromosome carrying capital Y will be coming in a gamete. So which is why all of them it is written as capital R, capital Y. Similarly over here also it's the same thing. It's small r, small y, whatever you do from each pair, you will be getting something like this in the gametes. So all the gametes are going to be same, my dear children. Now suppose this gamete and this gamete fuses. What will happen? You get something like this. From one side you get capital R, from other side you get small r. From one side you get capital Y, from other side you get small y. All of them will be in one particular zygote that will be found, my dear children. So can I just write capital R, capital Y, small y? The progenies that will be produced, my dear children, don't you think it will be capital R, capital Y, small y? Yes. So since capital R is there, it will be round, capital Y is there, it will be yellow. All of them are heterozygous, round and yellow. We started off with homozygous, but now we are getting heterozygous. Now again he did selfing, he crossed between the progeny, my dear children, and when he crossed between the progeny, this time the gametes were different. How do you find different gametes? Now can I erase it? Let's erase it and let me explain this to you. So this time it is capital R and yes, it is small r and capital Y and small y. I'm going in details like a kid, okay, because I know you might be needing it. I hope it's helpful. I'm not going way too much. So basically, from here again from a pair only one allele can go. Suppose this went. Now from here also one can go, either it can be capital Y or small y. So I wrote capital Y. Now again from this pair one can go, and from this pair suppose this time small y went. From here again I am writing small r went, and here again it can be capital Y, or it can be small r from this and small y from this. These are the maximum type of gametes that we will be getting. You won't get anything different apart from it, right? Anyways, you will be getting just such type of gametes. Now you cross two plants which were like this. So obviously two gametes which were like this. You are getting two sets of gametes which are like this. Now when you cross this with this, you cross this with everyone, okay? Capital R, you got capital I, capital R. Capital Y, you got capital I. Again, capital R, small r, so capital R, small r. Capital Y, so capital Y. This is how you have to make the Punnett square. Now what do you observe from this? Okay, you are never asked about the genotypic ratio. Okay, you are never asked about the genotypic ratio because there are so many genotypes up over here. But the questions come generally on different things. But genotypic ratio has not been asked for MCQs, for PU2 board examination. Again, they are not going to ask you genotypic ratio, but they are going to ask you what is genotype of round and yellow, what are the possible genotypes of round and yellow and things like that. So my dear children, this is round and yellow, this is also round and yellow. This is also round and yellow. So you have different genotypes of round and yellow. You should be knowing it. Just as a common sense, you should be knowing it. That's all. You are asked about the phenotypic ratio. So how do you find the phenotypic ratio? Basically, there's a very simple trick. 9 for the dominant parent that we had, that is round and yellow, 1 for the recessive parent that we had, that is wrinkled and green. Okay, round was with yellow and wrinkled was with green. Now you put 3:1. So here 3 and 3 basically are what are the new combination that you get, recombinants that you got, new combinations. So round was with yellow, but this time round will be with green, my dear children. You cannot put round and wrinkled together because this is the same character. It cannot be coming together. Okay, so you put round and green together, that is 3. Again you put what, wrinkled was with green, so this time you would put wrinkled with yellow, so that will be 3. So round green is 3 and wrinkled yellow is also 3. So 9:3:3:1 is the thing. Cool. This is Mendel's dihybrid cross. Cool. So you should be knowing about this ratio, very much important ratio. Now according to this, he put next law, that is the third law of Mendel, which is law of independent assortment. And what does this say? That when two pairs of traits are combined in a hybrid, segregation of one pair of character is independent of the another pair of character. And you might be like, ma'am, what? So basically, he was saying that suppose this is a condition. Okay, this denotes what the character that is shape, that is round and wrinkled. This is for the character that is the color. This is for shape and this is for color. Now he is saying when two pairs of traits are combined in a hybrid, segregation of one pair of character is independent of another pair of character. That means from this pair, capital R can come or small r can come. Either way, it's not like that. If capital R is going, then from here also capital Y will be going. From here, small y can also go with capital R. You get it? So it's totally independent of what will be going, and this is known as law of independent assortment, my dear children. They are independently, when you're talking about two characters, a pair of alleles, they will be basically independently assorting from each other. Okay, otherwise whatever you do, round will be, there won't be new combination that you can find. You are finding new combination. Round is with what's a green now. Three of the plants are round and green. Why? Because of these new gametes that are being formed, my dear children. You got it? And this is the law of independent assortment, and with that we have completed all the laws. Then and the exceptions also. Now that is done. So then the Gregor Mendel era, it went off. Okay, he did his experiment from 1856 to 1863 for 7 years. And after that came few people who rediscovered Mendel's work. So that is a straight question, MCQ question. Who rediscovered Mendel's work? So 3 scientists, De Vries, Correns and von Schirmack, they independently they were working and they rediscovered it in the year 1900. 1900, after so many years. Okay, now who said behavior of chromosome is parallel to the behavior of genes? So until now it was factors and things like that. There was no role of chromosome and things like that. Okay, so who actually said that basically the behavior of chromosome was parallel to the behavior of gene? So whenever I am saying that gene is separating from each other, basically it's not just a gene. It's the chromosome that is getting separated from each other, my dear children, because these genes are present on the chromosome. So this was identified later on. So in 1902, okay, in the year 1902, Walter Sutton and Theodore Boveri noted that the behavior of chromosomes was parallel to the behavior of genes and used chromosome movement. Okay, Walter Sutton and Theodore Boveri in 1902 said behavior of chromosomes were parallel to the behavior of genes and used chromosome movement, my dear children. Or basically just remember until here also, it's totally fine. Now, the next thing, what's the chromosomal theory of inheritance? So on the basis of this, when they did all this finding, they put down the chromosomal theory of inheritance. Who, my dear children? Walter Sutton and Theodore Boveri, they worked on it. But it was Sutton who put down the chromosomal theory of inheritance. So your book states that Sutton and Boveri said the pairing and separation of a pair of chromosomes would lead to segregation of pair of factors that they carry. That means, if these two chromosomes are getting separated, it will also lead to separation of these two alleles. Okay, that is the same thing that is being said over here. Now, on the basis of this, later chromosomal theory of inheritance were put forward by Sutton. Okay, it was Sutton who put forward it, but again you say Sutton and Boveri, Sutton and Boveri, Sutton and Boveri. If you have two options, Sutton and Boveri and then just Sutton, just put Sutton and come. okay, but mostly it won't be, they won't confuse you with that, but just understand. So who rediscovered? D. Rice, Correns and von Schirmack in the year 1900. Then some people came and said, hey, the genes that you're talking about, basically it's same like the chromosomes, my dear children. So it's what? Walter Sutton and Theodore Boveri in 1902. Then they gave the chromosomal theory of inheritance, okay, according to which same thing they said, whenever you're talking about a pair of chromosomes, when they are segregating, along with it something else is also getting segregated, and that is the factors which are on there. Cool? That's all. Now, going ahead, who gave the experimental proof to chromosomal theory of inheritance? Tomorrow if you do a research, no one will believe you. You have to do some experimental setup and show them. You have to give a proof. So who was the person who backed off and said that I am going to prove what the chromosomal theory of inheritance? It was Thomas Hunt Morgan, my dear children. Thomas Hunt Morgan. He was in the hunting phase. And basically, which organism did he choose for his experiment? He chose Drosophila melanogaster, or what you call it as? You call it as fruit flies, my dear children. And why did he choose it? Basically, because of all these reasons. They can be grown on easily on the simple media. It had a very short life cycle. So if I want to see some characters, if it's there or not, basically I don't have to wait for years. It has a short life cycle. It will die, it will get born. So basically it just was 2 weeks, that is a life cycle, and that was easy for me to understand what is the pattern of inheritance and things like that. Now, high progeny count. There were more progenies that were being produced. So he was able to see what are the differences in the progenies and things like that. Now, there was a clear male and female distinction. So it was not very difficult to identify which is male and which is female. So for pairing experiments and things like that, he needed that. Now, visible hereditary variations were there under low power microscope. So he was able to see the visible differences as well. So that was one major thing that Thomas Hunt Morgan used for his experiment. So which experiment is the chromosomal theory of inheritance experimental proof experiment. Okay. Now what organism? Drosophila melanogaster. And why? Because of all these reasons. Now it's not over. So basically what happened? Morgan only, he went ahead and he said that, okay, I see something kind of unique over here. And he said that there is something that is known as linkage, my dear children. Linkage is basically the physical association of genes. Didn't understand? Don't worry. So over here, suppose this is a chromosome. You have capital A, capital A. This is a gene over here. This is capital B over here. Again, you have another chromosome, small a over here, small b over here. This is a gene, this is a gene, this is a gene, this is a gene. Now, listen to me carefully, okay? Since they are far away from each other, even if these chromosomes come together and interchange certain things, it can easily make a new combination of certain things. For example, here AA is there still like this. Here capital B and small b is there because this part of it was removed and was being put over here. It's known as crossing over that was happening. So similarly, this part got exchanged here and this went that side. So you got new combination, my dear children. Why was this happening? Why was this happening? Because the genes which were over here, my dear children, they are not closely linked. They are not close to each other. If they were closely linked, probably both of them would have went together. There is no new combination that you will be seeing. But since they are very far from each other, so obviously which is why certain segments of it can be interchanged and you get new combinations. So what do you call these genes as which have longer distance between them? You call them as loosely linked genes. What do you call? Loosely linked genes. And when you have loosely linked genes, you see new combination, more amount of recombinations, new combinations would be found. Suppose this case was something like this. Okay, my hair. So it was capital A is here, capital A is here, capital B is here, and capital B is here as well. And from here they are going to interchange and do things like that. Basically they are close to each other. So whenever they are going to go together that side. You get it? They are going to go together that side. So that it is not going to bring much amount of changes. There will be changes but very less changes, my dear children. So closely linked genes, if you see, they have lesser amount of recombination. I am not saying there is no recombination. There will be obviously recombination, but the amount will be really less. So who gave this term linkage and all? It was Morgan who gave this term. Now closely linked genes means that they are very close to each other. If they are close to each other, they will be going together. There is lesser recombination. So you remember like this. If your friends are really close to each other, there is no chance another friend can come in that particular gang. So there is lesser chance of recombination, new combinations to occur. Okay, but if they are loosely linked, okay, my dear children, then there are higher chances of what? Recombination. Now, Alfred Sturtevant, okay, I always mess it around the name, Sturtevant, okay, he was a student. He was a student of Morgan, my dear children, and he said basically the frequency of recombination between gene pairs on the same chromosome is a measure of the distance between the genes. So if I say that the frequency of recombination is very much, that means the distance between them is also very much. That is what is said in simple words. So frequency of recombination between gene pairs on the same chromosome is a measure of distance between genes. Okay, that means if they are far, that means there is lesser combination, or basically if there is lesser recombination, that means they are very close to each other. Okay, if there is higher recombination, that means they are very far from each other. So basically you were able to do the gene mapping with the help of this, and this was being put up by Alfred Sturtevant. Cool, he was a student of Morgan, my dear children. So recombination, what is the highest amount of recombination? 50% is the highest amount of recombination, my dear children. So if you said a gene shows, two genes shows 37% recombination and another gene shows 1% recombination, what do you understand from that? If a gene pair show 37% recombination, another gene pair show 1% recombination, that means these gene pairs that you see over here, they are loosely linked. They are far away from each other, which is why there is high amount of recombination. Okay, and these guys which are showing less amount of recombination, that means they are what? Closely linked. This type of questions definitely comes, so go through it. 50 being the highest, obviously 37 is on this side, so it will be high. Cool. This is linkage and recombination, my dear children. Chalo, going ahead. Done? So this is a very important concept. So all the important concepts I am explaining over here, as I said, I am just repeating once more. I am not explaining everything that is there in your NCERT, just the important concepts which are being there. Now I might miss something here and there which I feel is not important, or from which I do not see much questions. But if I feel there is some point I have to put, I will putting in tomorrow's life. Okay, so do not worry about anything. Now, what are polygenic traits, my dear children? What do you mean by polygenic traits? So there are a few terms that you need to remember: polygenic traits, pleiotropy, and things like that. So polygenic name suggests what? There are so many genes. There are what? So many genes. So my dear children, usually whenever I say character and all, it's being controlled by a gene pair, that's all. Okay, a gene pair that is tall and dwarf, you have a pair of it, and that's all. A gene is going to control it. But over here, there are certain traits. Okay, polygenic traits means there are certain traits which are being controlled by multiple genes. For example, skin color and human height. The skin color and human height is a polygenic trait. So basically, each dominant allele adds to the trait while each recessive allele reduces it. So you can think something like this. So this is a pair. So here we have capital A, here capital A, here capital B, capital C, capital C. These three genes are going to together decide what is going to be my skin color, my dear children. So how many genes you have? 3 genes for one particular trait, that is skin color. So here also you have dominant and recessive and things like that. All the things that you see over here are dominant. So more dominant alleles are there, that means more darker skin will be there. So this will be coding for some protein, obviously giving a darker skin tone. Now suppose here one small allele, recessive allele is there. Okay, here one recessive allele is there, that means it won't be much dark. There will be little less lighter, it will be lighter skin color will be there. Not too much lighter, but little lighter than the dark one. Now again over here, suppose this is the condition, again more lighter. Okay, suppose this is the condition, again more lighter. Now you want to make it more lighter, again this is the condition, more lighter, there is no dominant allele over here. Same thing, more lighter. Same thing, more lighter. So how it is written? It is written like this. You will be asked which organism among them will be having the highest, which genotype among them will be having the darkest skin color. So darkest skin color combination would be like this: capital A, capital A, capital B, and capital C. This is a genotype. And what about the lightest one? Lightest one will be A, small b, C. If you are asked about, okay, choose, we have different options, right? So it can be like this also. So always choose for the lightest one, it will be all recessive, all of them will be recessive. Over here, this is a gene pair, this is another gene pair. So 3 genes are coding for what? Skin color. So which is why it becomes what? Polygenic traits. It is what? Polygenic traits. So many genes are there controlling what? One particular character. Example is skin color and also humanoid. Do not, I was saying do not forget. Do not remember it, oh my god. So do not forget it, okay? Now going ahead, there is something really important. So my dear children, have you already enrolled yourself into the Abhyas KCET test series that we have launched? Definitely, if you have not enrolled, you should be enrolling it. The offer price is 1,499. And basically with this 1,499, you will get access to PU2 Success Blueprint test series as well. So for 1 rupee, actually the amount was 1,498, but if you're adding 1 more rupee, you get the PU2 Success Blueprint test series as an additional benefit as well. So just avail it. It's a very important thing. As your KCET is coming, you can just go through all this chapter wise test and solution videos and one-shot videos and everything which is there. So the paper, past 5-year paper has been analyzed on the basis of this. This has been done. So it's a test series which you should be definitely taking. Do not take it in a lighter manner, okay? Just go and enroll, and the link to it will be given in the description box. So let's crack KCET with what? With utmost amount of confidence this time, and I'm sure you're gonna do that. Yes. Chalo. So polygenic trait was there.
Could you try again?
Okay, that was my Siri talking in between. Just let me tell Siri to not talk. I don't know why it spoke. Just a second. Okay. What are pleiotropic genes, my dear children? So pleiotropic genes is the ulta of the previous one. Basically over there, what did we have? We had multiple genes coding for one particular character, but over here there will be one gene that will be coding for multiple character. How is that possible, ma'am? I'll tell you. So the example for this is the disease that is phenylketonuria. Have you heard about it? It's coming under genetic diseases, okay? So phenylketonuria, in that what happens? There is a mutation in a gene that codes for an enzyme that is phenylalanine hydroxylase. What is it? Phenylalanine hydroxylase. So when phenylalanine hydroxylase is there, what does it do? It converts the phenylalanine which is there, which is an amino acid, into another amino acid that is tyrosine, my dear children. That is how it usually works, okay? But now, since there is a mutation in this particular gene that codes for this particular enzyme, obviously you cannot expect this conversion happen. In that case, what will happen? The phenylalanine which is there, it's not being converted, it just accumulates. And it accumulates as something that is known as phenylpyruvic acid. And this accumulation causes what? Mental retardation, my dear children. So this is one character that you can see. It causes what? Mental retardation. Because its actual work was to convert phenylalanine to tyrosine. That is the first character that is being shown by this particular gene coding for phenylalanine hydroxylase enzyme, right? Now there is also one character. When you're not converting it to tyrosine, my dear children, it indirectly hampers the production of pigments, that is melanin, okay? That is what? Melanin. So basically over here, there will be reduction in hair and skin pigmentation due to low tyrosine levels. So this is the second character that has been hampered. One was that, and the second is basically your skin and your hair pigmentation is being reduced. Please remember, it's not your hair is reduced. Hair is not going to be reduced. The pigmentation, the coloration will be reduced. So reduction in hair and skin pigmentation is due to low tyrosine level. This is the second character that is being hampered. So one gene, because of one mutation in one particular gene, this and this, both of them is getting affected, and this is an example of pleiotropic gene. Okay, this is an example of pleiotropic gene, that is polygenic, where multiple genes for one character. Pleiotropy, where basically one gene for multiple character. Cool? Chalo. Now we have sex determination, my dear children. So sex determination is different organisms. Okay, there are two categories. One is male heterogamety and the other one is female heterogamety. What do you mean by that? Heterogamety, that means different gametes are being produced. So when male produce different type of gametes, you call it male heterogamety. Didn't understand? I'll make it easy for you. So you have XY in the case of males. Human males, basically their genotype is X and Y. Now they can produce two type of gametes. One gamete can contain X, another gamete can contain Y. So you see two type of gametes are being produced, which is why you call them heterogamete. This shows what? Male heterogamety. Now it's not just human beings that do so. Drosophila also shows this. Drosophila melanogaster, the one that Thomas Hunt Morgan worked upon, right? That particular organism also shows X and Y type of sex determination, my dear children, where it is showing male heterogamety. Okay, cool. Apart from that, there is also insects, you know, grasshopper and all, they have a whole different thing. In them, the males are like this, where the males will be having just one chromosome, that is X chromosome, and there is no other sex chromosome, my dear children. There is no other sex chromosome. So, they can produce two type of gametes. One that carries X chromosome, another that carries nothing. But again, you have to consider it. Two type of gametes are being produced, right? So again, this is an example of what? Male heterogamety. Who are showing it? It's the insects. So you might ask, Drosophila is also an insect, but Drosophila is showing, my dear children, XY type of heterogamety. Over here, it is XO. This is XO type, this is XY type, and both of them are going to come under what? Male heterogamety. So in male heterogamety, you have given three examples. One is of insect, another one is of human being, another one exception is what? Drosophila. Now female heterogamety is kind of rare, but the females are producing different type of gametes. So in the case of human beings, we have X and X for females. So the gametes obviously will be same. But in the case of birds, okay, in the case of birds, they show female heterogamety, where males will be like this, ZZ. Both of them will be having Z, okay, or Z. Basically, that is the correct pronunciation. So the gametes produced will be same. Either way, that is male homogamety. Homo as in same gametes are being produced. But over here, the females are going to produce something like this. So either it can be Z or it can be W, my dear children. So it is showing ZW heterogamety. Cool? Works? So this is sex determination in both what you say as Drosophila, human beings and also insects, and that is about the birds. The birds, always remember, I don't know how to make you remember, but yes, they show this. Cool? Chalo. Now there is one different type of sex determination, my dear children, and that is seen in honeybee. So honeybee, they are always unique. So what happens is that basically all of us, if suppose we have 46 number of chromosomes, when something that is known as meiosis, when it happens, when gametes are being formed, okay, reduction happens and the number of chromosome becomes half, 23. That is the usual case that happens. So over here also, the females which is there, it will be diploid. They will be having 32 instead of 46. They have 32 chromosomes. Now obviously after meiosis happens, my dear children, at the end they will be giving you gametes that is having 16 chromosomes. That will be, if this is diploid, if this is the actual number, then half of it will be known as haploid. So this is female which is having diploid 2n number of chromosomes. Okay, it undergoes meiosis and it produces haploid gametes. Now in the case of males, they are already like this. They just have 16 chromosomes. So there is no point of halving it. There is no meiosis that is going to happen. There is mitosis. The cell will just divide into two and they will have multiple. Okay, for example, they are dividing into one more and it also has 16. That is, they are already haploid, my dear children. So from here it got 16, from females and also you have 16. Suppose this gamete and this gamete came together. 16 plus 16 is what? 32. And if it's 32, can it be a female or will it be a male, my dear children? It will be always a female. So diploid ones are always female. What about the haploid one? Suppose this was not fusing with anything. This unfertilized egg, my dear children, without fertilization, you will now call this as what? A male. So haploid ones, the half ones will be always male. Diploid ones always female. So this is haplodiploid sex determination which is uniquely seen in what? Honey bee. Cool? Works? Easy? Fun? Yeah. So we saw these many types of sex determination. All of them are equally important. Now to boost up your confidence and also for telling you the truth, I'll tell you, when I was a student, I also used to feel that this chapter is very much difficult. So many new terms are there. But trust me, this chapter is really interesting and easy if you know the concepts. So just focus on the fact that I am going to understand the concepts properly and I am going to solve as many questions as I can from this particular chapter. So tomorrow I will be having a fixed number of questions, probably 15 to 20, not more than that. but it will be a great help to you. In future, we will be obviously putting up more questions. I'm just putting 15 questions because I want to explain each questions with more priority, clarity. Okay, you get it? So if I just extend the live, it won't be of any use. We'll be putting it later on. First, you come and join the live. Then I'll talk to you. Yeah. Chalo. So now, disorders and pedigree chart. Now, this is something that gives you a short question every time. Even the female and male sex determination also gives you a short question every single time. So we have to be sure about all this thing. So disorders, if you see, my dear children, there are two types. One is Mendelian disorders and another one is chromosomal disorders. So when we say that a single gene is mutated, okay, one single gene which is located over here, this has some mutation happening, and because of this we are getting some disease. This is known as Mendelian disorders. Okay, so caused by alteration or mutation in the single gene, for example, hemophilia, cystic fibrosis, sickle cell anemia, color blindness, phenylketonuria, thalassemia, all of them, they are Mendelian disorders. Chromosomal disorders, as in when you're talking that, okay, one particular organism has one less chromosome or one extra chromosome, okay, or probably there is an alteration in the structure of the chromosome. This would lead to what? Chromosomal disorders. For example, you're talking about Turner syndrome when there is one less chromosome, Klinefelter syndrome when there is one extra X chromosome, Down syndrome when there is again one extra autosome. You get it? So there are extra chromosomes or there is a structural difference in the chromosome, which is why this particular disease arises. That is chromosomal disorders. Mendelian, as in you're talking about a gene and that gene is mutated. It can lead to all these diseases. Now, for understanding the diseases and all thing, you have something that is known as pedigree chart. And for understanding pedigree chart, you should be knowing the basic, right? So whenever you want to represent a male, you represent it by what? A square, which is unshaded, it's not colored, it will be plain square, okay, like this. For female, it is like round. For sex that is unspecified, you do not know the sex of the organism, it will be like this. For affected individuals, okay, you say if this is like this, that means this male is affected. If it's like this, then the female is being affected. If you want to show the mating, that a male and the female is mating, you show a line between them. This is what? Mating. If you see double line, that means it is mating between relatives. Okay, and you call this as consanguineous mating. Okay, what do you call it? Consanguineous mating. You call it mating between relatives, and this is something very important, comes usually. Two lines. Okay. Now, what does this show? That means male and a female is mating, and they are basically getting children, and one is male and the other one is what? A daughter, a female. Now, over here, male and a female is mating, but you are getting an affected son, my dear children. That is what you mean. Now, over here, it is mentioned like this. If you are not able to see, they have mentioned 5 and they have put like this. So, it's not specified, the sex is not specified, but there are 5 children which is being produced. That is what you mean by that. So, this is a flow of the pedigree chart, and if you analyze it properly, you will get it. So, questions directly come from this only, directly. What do you mean by this? So, you have to understand the diagram and tell that this is basically mating between relatives, which is why there are 2 lines. Cool? So, Mendelian disorder and chromosomal disorder, we are going to talk about it in detail now. So, under Mendelian disorders, that is where we are talking about the gene being hampered and things like that. So, there is sex-linked recessive disorder. What do you mean by that? Basically, the chromosome, sex chromosomes gene are being affected over here. The sex chromosome, in that there will be a gene and that gene will be affected, which is why they are getting this disease. Okay, sex-linked recessive disorder, there are 2 of them. One is color blindness, another one is hemophilia. What is the effect of color blindness? When I say that person is color blind, there is a defect in red and green cone cells. There is cone and rod cells. So, cone cell which helps in color identification, things like that. Okay, there is some defect in the red and the green cone cells, leading to difficulty distinguishing what? Red and the green color. What about hemophilia? I am going to compare and make you learn this. Hemophilia is where, my dear children, whenever we get a cut, that cut usually the bleeding stops, right? The bleeding usually stops. Why? Because in our body we have so many clotting factors which comes into play and just stops it. There are so many proteins which are there that helps in clotting. So, a single protein that is a part of a cascade of proteins involved in clotting of blood is affected over here. A single protein which is a part of very other different type of proteins which are involved in clotting is being affected over here, which is why it will lead to non-stop bleeding. So, over here person with hemophilia, if they get a cut, it will keep on bleeding. If a person has color blindness, he or she would not be able to understand the difference of color, different colors. You get it? Now, I say that color blindness is more common in males since they have only one X chromosome. And here also it said that passed from carrier females to male progeny. Not able to understand? I am going to explain to you really well. Listen to me carefully. So, suppose the female is like this, XX, and the male is XY. You know that, right? I am saying the female's 1X chromosome is affected. In that case, there is another X chromosome which is not affected. So, it will still overshadow the effect, and this lady is not going to have the disease. This lady is just going to be a carrier. If this lady has a disease, then the condition should be like this, when both of the alleles or both of the genes on both of the chromosomes should be affected. Then I will say that this lady is what? Diseased. On the other hand, if we talk about males, if one X chromosome is affected, there is no other X chromosome to overshadow the effect, and hence this person will definitely have what? The disease, whatever you do. So, which is why I say males anyway have one X chromosome and there is no one to overshadow the effect. So, obviously their chances of getting the disease is what? More. So, both color blindness and hemophilia is like that. Okay, both color blindness and hemophilia is that, where it is more common in males, 8% more, okay, since they have only one X chromosome, my dear children. Cool. And over here, females are usually carriers unless they inherit two defective copies. If they are getting two defective copies, my dear children, that's when they get the disease. Now, probability, a carrier mother has 50% of chancing it to her son. A daughter will be colorblind only if the father is affected and the mother is a carrier. Now, let's break it down. Okay, let's break this particular thing down. So, over here, shall I just erase it? Yes. Listen to me carefully. A carrier mother means it's like this. Only one chromosome is getting affected. Has 50% of chance of passing it to her son. Suppose the male she is marrying, it's normal. He is normal. The gametes that will produce will be X and the other one will be XC. Here the gamete will be X and the other one will be Y. This and this fuses, you get XS. This is a normal female that is being produced. This and this fuses, you get XY. This is a normal male that is being produced, my dear children. What about this and this? This is a carrier female because there is one X chromosome that is affected. What about this and this? You see, the male over here is definitely affected, whatever you do. Okay, the male over here will be definitely affected. So, carrier mother has a chance, okay, has a 50% chance of passing it to her son because of this particular case. Okay, so if this is a condition, it will be like this. Cool? Understood? Okay, so one child obviously will be normal, but the other child, if you see, he will be affected. Here one girl child will be carrier, another girl child will be normal. Now, a daughter will be colorblind only if father is affected and the mother is a carrier. Daughter, when here you see daughter is not affected anywhere. So, when do you think a daughter will be affected? Actually, this is 25% chance, there is not 50% chance. Wait, it's wrong over here. Okay. Now, if you are talking about daughter, I am saying the mother is a carrier, that is being said now. Daughter will be colorblind only if her father is affected and mother is a carrier. Yeah. So, now let's make the father. Anyways, one X chromosome has to come from the father, that's when the daughter will be affected, right. So, X over here, XC over here, again XC over here and Y over here. This and this fuses, you get X, XC, here she is a carrier. This and this fuses, you get X and Y, it's a normal male. What about this? This will be XC, XC, it will be a child who has what? Color blindness. What about this and this? It will be another child, but this is a male child who will be colorblind. But anyway, if you see, the chances of males getting affected is much more. Okay, How much chance is there? Basically 25%, 25, 25, 25, 25. 25% chance is there for a male to be colorblind, my dear children. Over here, what's the chance of a female to be colorblind when this is the condition? Again, 25% of it. What's the chance of a female to be, what say, carrier? It will be again 25%. What is the chance of a male to be normal? It will be again 25%. Cool? Works? Okay, so this is about color blindness, and the same thing goes for hemophilia as well. Same thing, bacha log, same thing. Okay, so passed from carrier females to male progeny. So suppose the female is a carrier. Now you just replace with H. Okay, and the male is like this. From male, if you want to make a son, obviously this guy has to come. And that is not affected. So if you want a male to be affected, from here, one affected chromosome should be coming in. That's when you get it. So how do you get it? So this and this fuses, you get XX. This and this fuses, you get XY, normal male. But this and this fuses, you get XHX. This is a carrier female. This and this, you get XHY. This is what? An affected male, my dear children. So you get hemophilia. Okay, a person will get hemophilia. The male will get hemophilia if his mother is what? A carrier, my dear children. Okay, why it is not said the mother is affected? He will also get when the mother is affected, because, my dear children, it won't, the survival rate is very less. The survival rate is very less. Obviously, they won't be living too long to reproduce and things like that. So anyways, let's make, okay. So over here, XH, X and Y is there. Fuse this with this, again you get X, again the female is a carrier. This with this, XHY, the male is affected. This with this, XH, this is again a female carrier. This with this, again the male is what? Affected, my dear children. So both these cases the male is going to get affected. So it is generally said the males passed from the carrier females to male progeny. Now females rarely affected because it requires an affected father. So females will be affected, my dear children, when there is an affected father and there is a carrier female. When there is a carrier mother, okay, like this, and also an affected father, that's when the female will be getting affected, because this and this, by chance if it comes together, there will be something like this, and that's when she will be affected. You get it? So for females to be affected, the mother has to be carrier and the male should be affected. That's when you get this. Now, Queen Victoria was a carrier of hemophilia. So I hope you understand sex-linked recessive disorder. It is totally linked with the sex chromosome, which is why it is known as sex-linked recessive disorder. Why? Because it is only expressed when both of them have the chromosome like this. Okay, when it's in this condition, either way, either should be like this or it should be like this when it should be expressed. There is no other one dominant to cover this up. That's when it is being expressed. It's in recessive condition only. Cool? Now, next, you have sickle cell anemia. Now, you have autosome linked recessive blood disorder. What do you mean by autosome linked recessive blood disorder, my dear children? So, basically, you know, we just have a pair of sex chromosome. All the other chromosomes are known as autosomes. What do you call them? Autosomes. Pair of sex chromosome, you call them allosomes. And all the other pairs of normal other chromosomes which are not linked with sex, they are known as autosomes. Okay. Now, this is a defect which happens when a gene in the autosome is getting affected. And the first disorder is sickle cell anemia. Now, sickle cell anemia, I'll call it, it's a qualitative blood disorder. That means over here, the quality of something is getting hampered. Let's see what. So, the effect is mutant hemoglobin changes RBC shape to sickle cell, leading to reduced oxygen transport and blood flow obstruction. So, over here, there you know in the RBC you have a pigment that is hemoglobin, and hemoglobin is made up of different proteins, that is alpha globin chain and beta globin chain. So, over here, in the case of sickle cell anemia, the gene that will be coding for beta globin is affected. The gene that codes for beta globin is affected, which means you are indirectly affecting the HB, that is hemoglobin. The structure of hemoglobin is basically affected, my dear children. Okay, that means it is going to affect RBC also. Okay, when there is a mutant hemoglobin, it is going to change the shape of RBC, which was something like this. It is discoid in shape to something like this. Now, in this shape, it won't be able to carry much amount of oxygen. That is one thing. Second thing, it might obstruct the blood vessels as well, because we know the shape is also very, very important. You get it? So that is a problem. Now, it is caused by mutation of beta globin chain. I told you, it is caused by mutation of beta globin chain. So this is a normal chain, my dear children. Okay, this is a normal chain where you have HbA gene. Okay, you have Hb gene, which is basically going to help in the formation of what? The beta globin. So over here, the DNA was CTC and GAG. This is a 2 double-stranded DNA strand that you're seeing forward. You're looking over here. Now over here, only one strand is transcribed to form an mRNA. So from CTC, you get C makes a complementary pair with G. T, it makes A. From C again, you get G. This is the mRNA. You don't transcribe both the strands, you just transcribe one strand. So this is the one strand that is getting transcribed. In that case, the amino acid that will be produced will be what? Glutamic acid, my dear children. It will be what? Glutamic acid. Cool. This is a normal amino acid which will be produced. But over here, when there is mutation, okay, when there is a mutation, what happens is that it is CTC over here, but it has become CAC over here. So if CAC is there, then it will be pairing with G, U, G. So when this changes, when this mRNA, my dear children, it changes, what will happen? Again, from mRNA's codon, what do you get? You get an amino acid. Each codon codes for amino acid. Here GAG was coding for glutamic acid, but now GUG will be coding for something else that is valine. And this is going to bring out the whole change. So this is caused by a mutation in beta globin gene, where glutamic acid is replaced by valine at the position 6 due to GAG to GUG substitution. Where it is happening? At the 6th position, my dear children. So you have a whole chain. You have different codons, 3 codons, 3 codons, 3 codons, 3 codons coming together. So I am saying in the 6th position, okay, this change is happening in the beta globin gene. Okay, in the beta globin, the gene that codes for beta globin. Okay, now when do you say that this particular person has a disease, my dear children? It's a recessive condition. That means both the genes. Now again, you should be having, okay, so just a second. Over here, if both the genes are affected, that's when this mutation is going to happen. Okay, or basically when I say, not mutation is going to happen, when I say both the genes are affected, both the over here both the genes are getting mutated, that's when the person is going to get diseased. You say that that person has sickle cell anemia. When the condition is like this, where you have still one allele which is normal, HbA, and the other allele which is affected, that is HbS. In that particular case, since there is a normal one, it will dominate, overshadow its effect, and that person will be just a carrier. That person will be just a carrier, my dear children. Suppose in the next condition, both of it is having HbA, that means it's a normal condition which is there, normal cell. Okay, so when both of the alleles are same, homozygous condition is coming over here, that means it's a diseased individual that is going to be formed. Heterozygous condition is going to form, that means that will be a carrier, my dear children. So over here, do you think that males have more probability, females have more probability? Nothing like that, because it is autosome linked genetic disorder. And over here, the quality of RBC is getting reduced, which is why it is known as qualitative blood disorder. Why I am stressing on it is that you have 2 disorders to learn. One is 2 blood disorders to learn. One is thalassemia, another one is sickle cell anemia. Sickle cell anemia is messing up with the quality, and this question is being usually asked. Over here, the codon GAG is being replaced by GUG. There's only one particular thing that is being replaced over here. One, what do you call it? A nucleotide. Okay, one nucleotide which is being replaced over here. Always remember, A comes before U. So previously it was A, and then it was converted to what? U. Cool. Similarly, here also G comes before V. So glutamic acid was the first thing that was being coded, but now valine is being coded, and this is for beta globin chain. Whereas beta globin gene produces, my dear children, this particular gene is found on chromosome number 11. It didn't mention over here, but I'm telling you it's chromosome number 11. Probably that particular chromosome is getting affected over here. Got it? Chalo. Next thing, another, okay. Autosome linked recessive disorder, again the blood disorder, I told you thalassemia. What happens in thalassemia? So basically two things can happen. You can get either alpha thalassemia or you can get beta thalassemia. What do you mean by alpha and beta thalassemia? So I told you there is alpha globin chain and there is beta globin chain. So gene coding for alpha globin chain can be affected or gene coding for beta globin chain can be affected. Either way. Now in the case of alpha globin chain, this is the condition. They have two genes. One is HBA1 gene, another one is HBA2 gene. Okay, allele for this is this and this. Allele for this particular gene is this and this. A person can get all the alleles to be affected. In that case, I call this as a thalassemia major patient. Okay, thalassemia major. That means in their body, alpha hemoglobin or alpha globin chain is not being produced at all. At all. Suppose two alleles over here affected, but you have still two alleles which are normal. So I will say still that person has thalassemia, but in their body not too much amount of alpha globin chain will be produced, but still it will be produced, but not too much. So I will call this as minor condition. So this is again, you are talking about quantity of globin chain which is being produced. If all of them are affected, that means it is you're not going to produce anything at all, that is globin chain at all, alpha globin chain at all. But if one or two alleles are affected, that means you're still going to produce but very little. So there are, always remember, there are two genes and two genes, HBA1 and HBA2. Two genes have two alleles also. So 4 alleles are there for this particular thing, and this is on chromosome number 16. It is what? Chromosome number 16, my dear children. When you talk about beta thalassemia, it's on chromosome number 11 we just studied, and there is only one gene having 2 alleles. Okay, either 2 alleles will be affected, that means there is no beta globin chain that will be produced, or one allele can be affected, that means some amount of beta globin chain will still be produced. So here it is a quantitative disorder. Why? Because of the reduced synthesis of alpha or beta globin chains. It's a quantitative disorder. Got it? This is again autosome linked disorder because you're talking about the 11th and the 16th chromosome. Okay, the sex chromosome are the 23rd pair of chromosome. Okay, they have nothing to do over here. Now there is one more autosome linked recessive disorder. Okay, so we studied 2 disorders which are sex linked recessive disorder. Then we studied about 2 disorders which are autosome linked recessive disorder, which are blood disorders. Now there is a third autosome linked recessive disorder, but this is not a blood disorder, this is a normal disorder, that is phenylketonuria. Okay, phenylketonuria, we already studied about it, that right. So here lack of phenyl hydroxylase enzyme is there. This particular gene, the gene that codes for this particular enzyme is not being there. Okay, it's not like it's not there, it's basically getting mutated. Okay, it is getting mutated, which is why the tyrosine, phenylalanine to tyrosine conversion does not take place, and in that case what will happen, the phenylalanine and its another version, that is phenylpinedoic acid, will get accumulated and it will lead to mental retardation. Now there is one very important symptom that is which you have to learn is excretion. So basically the people who are having phenylketonuria, they have excess compounds that are secreted in urine due to poor kidney absorption. So that will be too yellow in color. Okay, it will be too yellow in color because there are certain acetones phenylacetones that will be finding there. Okay, you will be finding ketone crystals in the urine. So excess compounds are excreted in the urine due to poor kidney absorption. Okay, kidney won't be able to absorb, so obviously all these things will be defecated out. Got it? So this is phenylketonuria. All these diseases, my dear children, are Mendelian disorders, gene disorders. Okay, the gene is getting altered over here. Now we are going to talk about chromosomal disorders, but again before that we were talking about recessive disorder, but now we have something that is not recessive, but why I have put over here again, I don't know. Anyways, there is something that is dominant. Example is given, and there are 2 examples of pedigree chart also, which is something that we need to study. So how do you understand what is dominant and what is recessive? So autosomal dominant trait means, for example, the condition was like this, where you saw that this was there to dominate and cover up the situation. But in this case, even if one is affected, okay, suppose this is affected, still it will dominate and this particular person will also have the disease. That is autosome dominant trait. Example of this disease is myotonic dystrophy given in your NCERT below the picture. This picture, no, and below that it has been given. So it is autosomal dominant trait disease, my dear children. And these diseases, how do you identify? If you are asked which is autosomal dominant trait disease and which is autosomal recessive trait disease, how can you identify it? I'll tell you. Basically, in the case of dominant traits, okay, every generation you will be finding some person who is deceased. So in the first generation, the female was deceased. In the second generation, the son and the daughter is being deceased. This is in the next generation that is there, another organism is being diseased over here. This is what, and from one pair you will be finding at least one person to be diseased. Okay, from pair you will be finding at least one person to be diseased, that is autosomal dominant trait. So at least one trait, if it's there, it will be passed on to the next generation, however, somehow. Okay, but in the case of recessive disorders, my dear children, such as sickle cell anemia and things like that, it will show alteration of generation. You won't find in every generation, not alteration of generation, basically it will be missing certain generation. I don't know why I mixed botany over here, but yeah. So in this generation, this disease is not seen, no one is affected, but here the disease is seen. And again, these parents do not have it, but again these children, they have it. So there is what, they are skipping the generation. Why? Probably they might be just carriers. And when two carriers come together, you get something like this. Get it? So it is autosomal recessive disorder. You can track it like this. And here the sex is not being specified. So it cannot be sex-linked disorders, my dear children. Cool? So this is the pedigree chart that they can probably give you, nothing apart from it. Now, talking about chromosomal disorder, which is the last part of this chapter, my dear champions. So let me just see the time. Yes. So chromosomal disorders, again you have two. One is aneuploidy, it can lead to aneuploidy, and the second one is polyploidy. Not it can lead to, basically there are two types or two causes of chromosomal disorders. Aneuploidy is basically when the chromatids, you know, they have to separate from each other when they are forming gametes. Suppose they are not separating, and the gametes, both of them are going. So basically you see addition of chromosome is there. Okay. or there can be loss. Okay, I went over here, these chromosomes went over here. What about that gamete? That gamete is not having any chromosome, so there is a loss of chromosome over there. This is known as aneuploidy. What do you call this? This is known as aneuploidy. Very, very important term, my dear children. So failure of segregation of chromatids during cell division cycle result in gain or loss of chromosome. It can be either gain, it can be either loss, and this is known as aneuploidy. What do you mean by the next thing? Polyploidy. Polyploidy means failure of cytokinesis after telophase stage. So what happens is that there is a cell, okay, this cell will be dividing into two once its chromosome and everything is there. But over here, there is no cytokinesis, the cell is not dividing after the telophase stage. In that case, what will happen? There will be a whole increase. Suppose it had 23 over here. Now since the cell is not dividing, instead of having 23, what is this thing having? 46. So that is a huge problem. So there is a whole set, increase in the whole set of chromosomes together, my dear children. And this is often seen in plants. This is not seen in mostly animals, it is seen in plants. This is known as polyploidy. You would be asked mostly about aneuploidy, my dear children, where there is when you're talking about each chromosomes, a pair of chromosomes, it is not segregating properly. And that will lead to loss or gain. This is known as aneuploidy seen in us. Now, when I said when there is an additional copy of a chromosome, you call this as trisomy. You call this as trisomy. Why? Because listen, from one gamete you are getting one chromosome, from another gamete you should be getting one chromosome. Together it will be something like this. But now over here you have one more chromosome up over here. So what are you getting? You are getting one extra chromosome. So 1, 2, 3, trisomy is happening up over here. Then there is something that is monosomy. So individual may lack one pair of chromosome. So from here there is nothing that is coming. From here you are getting one chromosome. So if you see, there is only one chromosome that will be there in the cell. There is no other chromosome. So this is known as monosomy. There is only one chromosome for a trait. Okay? Cool? So trisomy and monosomy comes under aneuploidy. Yes. Chalo. Now, you have different conditions in aneuploidy. I told you trisomy, monosomy, everything will be explained up over here. You have 3 diseases, and let me give you the good news. This is the last slide of this particular chapter. So we are probably going to complete this chapter within 1.5 hours. Do you see the deal? It's not a small deal. Great. So I hope if you were there with me until now, you should give a very nice tap on your back and say that, hey, I did it. So yes. So my dear children, Down syndrome is caused when you have a trisomy at 21st chromosome. That means You get a normal chromosome from one parent. From another parent, you're getting 2 chromosome, and this is the 21st chromosome. Okay, so from one pair you're getting this chromosome. This is, suppose it comes together, and this is now a 21st pair of chromosomes. Okay, but over here getting one more extra. So what do I call this? This extra chromosome is there. Okay, extra 21th chromosome is there, my dear children. But over here we usually say the condition is something like this. We have 44 autosomes, my dear children. Okay, we have 44 autosomes and plus XY, or basically XX chromosome, that is 45th and 46th chromosome. In this condition, why I don't know it's 47, wait, it's wrong. It will be 45A. So instead of having 44 autosomes, you know 21st chromosome is what? An autosome only. I told you 23rd is a sex chromosome, so there is nothing happening to sex chromosome. There's an additional autosome, which is why I am writing 44A plus XX, or it can be XY. So if it is XX, then it is a female who is having Down syndrome. If it is an XY, then it is a male who is having Down syndrome. Features, what are the features? Short stature will be there, round head will be there, furrowed tongue will be there. The tongue will be looking like this. There will be a furrowed tongue. Okay, broad palm crease will be there. Mental retardation and delayed development will also be there in Down syndrome. Remember, this is an example of aneuploidy, since there is a gain of I went to chemistry, sorry. So there's a gain of chromosome up over here, and this is an autosome, not sex chromosome. So which is why instead of having 44 autosomes, here this person is having 45 autosomes and can have either XX or XY, my dear children. Cool. Now coming over here, talking about Klinefelter syndrome. What is the case over here? So usually from father or mother, basically X, and from here you're getting Y. Okay, but here the condition is that instead of getting just an X and Y, you're getting an extra X chromosome. You're getting an extra X chromosome. Now it can be from here, it can be from the father's side, whatever you have. Okay, but anyways, you're having an extra X chromosome. So how many autosomes are there, my dear children? Autosomes, if you see, 44 autosomes are there, but the condition becomes what? XXY, an extra X chromosome is there over here. So you can write either like this, or you can write 47XXY. Either way we can write. Most correct will be this one. Okay, because there is 47Y, my dear children, 47, one extra chromosome is there, which is Y7, 47XXY. Cool. Now over here, listen to me very carefully. It was supposed to be a male. Okay, so there will be masculine appearance for sure, but with gynecomastia, that means there will be development of breast as well. This person will be sterile. It's not fertile person. Okay, it will be a sterile person. They won't be able to give rise to offspring. Now there will be incomplete secondary sexual development as well in the case of Klinefelter syndrome. So again, a masculine body, okay, with gynecomastia, there will be development of breast and everything which is there. This is a chromosomal disorder where there is an extra X chromosome that is coming in, which is why it becomes 44A autosomes plus XXY, or you call this as 47XXY condition. Okay, where you are not referring to autosomes at all. Now that is known as Turner. There is something else that is known as Turner syndrome. What happens over here? So there is a missing X chromosome. There is a missing X chromosome. So instead of having XX, there is only from one parent you got X, from another parent you didn't get anything, which is why it is just one X chromosome which is there. Okay, so again it will be 44 autosomes plus what? Plus X. There is nothing else. There is XO, you write it. Okay, there is nothing else there. Okay, or probably you can write it as how many total number of chromosomes is there? It is 45. And what about the sex chromosome? There is not one. There is only one. There is no other one. You get it? Now what happens over here? It's a female, but the ovaries are rudimentary. It will be sterile. There will be lack of secondary sexual traits, and the stature will also be short. This is known as Turner's syndrome. So apparently this had to be a female, but what do you have? You have rudimentary ovaries and things like that. There will be no lack of secondary sexual characters as well as short stature. And this is supposedly male, but it has to be like this. So these are chromosomal disorders, and these are the examples of aneuploidy, where you're talking about gain of chromosome and there is a loss of chromosome, my dear children. And with this, we have completed the chapter that is what? That is principles of inheritance and variation at the last. And I have got a little homework for you. I told you that there is something which is an example of polygenic trait as well as multiple allelism. Right? Where did I tell? After your blood group thingy, you find it. Can you just tell me what is that example about in the comment section below? If you have heard me until now, you should do me this favor and do let me know about this. Then only I'll get to know that you heard me, and also you are taking things really, really serious. So it's not like any ma'am will come and teach you and go and you won't put your efforts. It should not be like that. You should definitely put your efforts. Okay, if you're not doing so, it will be a huge trouble. So please put your efforts, and we will also put our efforts and be there sharp at 11 a.m. tomorrow for the live. And if you enjoyed this thing, if you have any doubts, ask me right away. Do not wait for anything and not feel that you're going to be judged, you're on a social media, things like that. No one is able to see you, my dear children. And even if people are able to see you, what's the problem in asking the doubt? Doubts brings out discoverers. So have that thing in your head. Okay, so that's all, and do not forget to like, share and subscribe. Bye bye, take care.